So we will stay together in these few minutes, and we will see what is possible, which technique are possible to use to study solid state in drug development. So at first, I'm happy to be with you, all of you, and to talk. So if you have questions at the end, you can ask, or you can write me by email, or for future collaboration, why not? So what we're going to see, well, actually, I try to, uh, uh, yeah, okay. So what we're going to see, we're going to see the solid state, and solid state can be modified, heating and cooling during preparation of drugs, and also crystallization can perform a solid state. And what we see also polymorphic search, because solid states strictly connected with new polymorphs, not only polymorphs but also called solvate, co-crystal, and crystals. And we will see all the analytical technique for solid states, more or less, not quite all, but a lot of them. Why? Well, I study crystal solid form, because it's very important for therapeutic effect, for the end of the drug, and usually the phenomenal crystal prediction is connected with serendipity. You can start when you synthesize, and you do really the different steps, and at the end you have a pure form, and from that point it starts um, a new crystallization with a really different technique, and new drugs can appear, and so new crystals also. And this, why is this so important, this solid form? Because the solubility and bioavailability can be different of a drug, from one form to another, and also the hygroscopy can alter the therapeutic effect. So it is important to study the property, the chemical and physical properties of the solid forms, and to study also how they have been they are going to be prepared inside the pharmaceutical form, and then to do a map, not only to study the reagent at the beginning, but to doing a map of all the path of the solid forms of the crystalline and not crystalline phase, because sometimes are not crystallines and are amorphous. So it's important from the beginning of the reagent, during the formulation, during greening or eating of solid forms, and also during stocking of the new drugs. So what we started once upon a time, and so we decided to start it at first to study polymorphism, because one substance, a pure substance, can modify its structure in different structure and create different polymorphs, and so different therapeutic effect of the drug. So you can understand how it's important to have that drug and not the other one, not a mixture. So it's important to characterize each solid forms. And it's important to characterize with different technique and to study the chemical properties not only of the polymers, but also of the hydrates, solvates, amorphous, and so on. And we will see which technique have been used for this or that or the other one. Well, actually, in the lab, we have a lot of them. And so we have applied in these years a lot of techniques to study on them. So what we have applied, so we will see together different examples of solid form study with different technique. For example, the SC, it is calorimetric differential scan. TGA, I'm not going to show the results. And with Raman and IR, two spectroscopic two technique that are complementary techniques. So one is connected with polarization is Raman, and with absorption it is IR. And we have studied also with X-ray diffraction, with X-ray diffraction, with powder diffraction, and so on. Um, and so is we study also not only at room temperature, but also during the grinding, during the eating of the solid forms, and also during the crystallization. So I'm going to show you some example of this, what we've done. So as I just told you just few minutes ago, it is important to study. Well, no, I'm doing like that because okay. It is important to study the solid forms from the beginning during the preformulation. Preformulation means that is to study the reagent, to study the polymorphs, the eventual polymorphs that are going to appear, and to study the online monitoring, and it's possible to study with also with portable instrumentation, supporting chemical and development process scale up. And it's possible to study in qualitative and I say semi-quantitative analysis of the active substance, but not only of the thick substance of the, but in the tablets, in the powders, the liquids, emulsion, gels, vials, and capsules, transdermal and transdermal drug delivered pads. It also all the other excipients. So, because the excipient can cause the modification of one forms to another. So it is important to study also the excipient during the formulation, so during the greening, heating, and so on, but also it is important to study the final forms during the storage, during the monitoring of the drugs. So, what we had done, after we started with polymers, but also we studied crystals and salt and amorphous solids. For example, for the amorphous, it's Raman technique can be used, and you know that X-ray is not the best technique to use for amorphous. So you have to decide also which technique use, and which is not better used for that in that case. But returning to the analysis of EPI, why they are so important is to study their unique physiochemical properties can modify bioavailability, and also manufacturability, purification, and also the stability and the performance of a drug. The performance of the drug is directly connected with the therapeutic effect at the end. So we studied at the beginning three different AP. So we studied with three. The first one was chloroform palmitate. You can see here the structure. I'm not I just ran not show you the synthesis as well, not the synthesis, 'cause the crystallization. Because the crystallization is very important when you study polymorphs, and you have to crystallize really in different way, and to try also different crystallization. And usually this crystallization are patterns binded with patterns. So, for example, in this case, this form where form A was a stable form, and it is inactive, biologically inactive, and B is a metastable form. It means that can modify itself because of temperature not only, and it is an active modification, and C, it is an unstable form. Both of them, all three were not patent, but usually are patent. Polymorphs, usually you know how to crystallize one or two polymorphs, and the other are protected by patent. Why? Because, as I told you, they are connected to therapeutic effect, and so pharmaceutical firms which crystallize one polymorphs, it means that the structure is like that, and you can crystallize in three different polymorphs, for example. But the formula and is like that. It means that, for example, A is inactive, and the other two modifications, B and C, are active. But one, it is an unstable. The other is metastable. So you have to study in which condition the form B is metastable, and not have a final drug with A and B and C, a mixture, and have one of these drug active. But you have to study at first, at the beginning. So this is the reason of mapping all the production of a drug, a new drug, and this is the reason, the region how why the pharmaceutical firm put the patent on each polymers. You have to study how, for example, metastable form B active modification can transform itself during grinding or eating or something else, or cause of the pressure, for example, because it's not possible to have a final drug with an active modification, for example, form A, or with really an unstable form C. So, which kind of technique we might use? Well, actually, not only we. When you do characterization of solid forms, you have to use not only one, but you have to use different technique for characterization, identification of the drug. Because I've told you, it is a the structure, the molecular structure is not modified, but what is modified? Modifies, we will see, we modify some ligands or the crystalline forms. So you have to use, for example, FTIR or micro IR, or you can use a Raman, micro Raman. In this case, we have used FT Raman, or you have to also to study the solid forms during eating. So if you study during eating, you have to use a part of the instrument which heat the samples, and it's called hot-stage Raman microscopy, and also other technique. Or you can study with other technique. The other technique are not destroying the samples, more or less. Well, it's actually the outstage, yes, but the other no. This DSC is different scanning calorimetry is possible to use and to study during eating the solid sample, and it's possible to use also X-ray diffraction, powder X-ray diffraction, or single X-ray diffraction. We use both because the crystals, when they are three, four, or six, I don't know, polymorphs, the crystal, so the three-dimension A, B, and C are different. So it's not surface, and if the sample is made of it is crystalline sample, it's necessary to use also single X-ray diffraction. What we do, what actually in this case we have used, we compare all the bands, but not just to say is present or not. We study the similarity and what different one form to another. So these are reported FTIR in this column from A, B, and C, and here FT Raman from A, B, and C, and then we at the end we put also the segments. So we study, for example, that the forms you can see that they differ really for few bands. So really, for example, VS means very strong. This is a medium. This is a strong and medium. But also we study also very weak band to see if very weak like this, very weak, if there are some difference. So we could differentiate. We could in this case we could crystallize three different polymorphs, and we could also differentiate the three polymorphs with FTR and FT Raman, not only for the strong band but also for the weak and very weak. And then we assign and we do assignment of the peak. So we compare in this case FTIR from the up with FT Raman to the down. And just to have an example of the three. And also it's possible to cut, for example, here to cut, to have a strict range of the three forms. In this case, A, B, and C. This is after Raman. And so after Raman, as I say, Raman are bands from the down to the up. And FTR from up to down, and we could remember that A is an active and it is stable, B is metastable and it is active, and C is unstable and active. And also we could finally we could compare, and we have tried also a mixture to do a semi-quantitative analysis of the three forms, because when you crystallize in a small scale up, you're sure to have well, sure, yes, you're sure to have one form and not the other, but sometimes it's possible also in a big scale up to have a mixture. So you need to identify, categorize also the mixture, and so we have tried to have a mixture of little percentages of one form to another, and to see if we could see the differences, and it was possible, and also for the subtraction. In the other case, we have studied tamoxifen citrate, and form 1 and 2, who you can see here, they were well known. Have not reported the crystallization of these. Are all these published? Have not reported the crystallization of the forms. Remember, this is important point, the crystallization. So from 1 and 2, they were well known, and for 3, no, it was a patent. Okay, so it was a must to, well, actually it was quite hard to do crystallization of this, but we could do it. And then we have done the identification categorization with our bands of all the three firms, and we have identified which band could differentiate one form to another, and also we have done the segment. We do like that. So each time it's not only is present, not present. Look, the different. Yes, you say, oh, they're different, not too much. Well, for us, they're different a lot because, for example, after ramen and micro ramen, they have a high resolution. So, differences are important also from this to the other one. So one is medium and one is weak, and also one is also, if you see, oh, it's just a number, the fourth number, okay, but for us is a big difference from one to another. So we could We could study with a TIR and a TIR. In this case, we have compared all the three forms, so 1, 2, and 3. This and also we have done the comparison of the three forms with all the assignment. Is it easy to do? No, because you can find a database, yes and no, because you know, if one polymorph, for example, is plotted by a pattern, you have to know how to identify categorize, and it is supported by a patent. You have to know very well, in this case, spectroscopy, FTIR, micro-IR, and micro-Raman to assign. In this case, we have a study during the crystallization of the sample. Tamoxifen citrate was put inside a melting point tube, and we have done Raman experiment, and the Raman is connected with the computer and so with a camera, and it's possible to have an optical image of the samples. And so we have realized at the beginning it was form 1, then it was form after 44 hours it was form 2, and then at the end after 48 and 50 hours it was form 2. To say what? That you also during customization, when forms can modify itself to another. And so you need to identify and crystallize also, identifying and crystallize also during the crystallization. And in this case was done through the holders, through the glasses. With Raman is possible because the glasses, it doesn't give a Raman signal. Well, actually, I say like that, but it's not truly, but it's quite. And this, and just to focus your attention that with micro Raman it is possible to study through the holders. It's made of glass, and also inside with water objective immersion. Micro Raman is instrumentation which is composed by by objective. In this case, it's possible to put the water immersion objective inside the solvent, and the water is a weak Raman scattering. Water, it doesn't give a big signal like in IR, and also organic samples and everything is present inside water and also in organic sample. It is possible to study through the holders and with the water objective immersion. In this case, also we have used X-ray diffraction. After X-ray diffraction, powder X-ray diffraction for form 1, 2, and 3. Yeah, because it was crystalline, and we have used also the SC and eating the sample. In this case, yes, it destroying the sample. The other technique are not destroying. Well, actually, if you eat the sample, yes, you modify and destroying the sample, but the spectroscopy technique are not destroying the sample. So we have study in this case with the SC, and we study how it was modifying itself during eating. It's very important also during the production of a drug to understand what's going on during eating. Then we have started to study acetylazonamide, and we have started to study form A and B, and to compare FTIR with microraman. As I say, what IR can see. Look, here, what I can see, Raman not, and vice versa, are two complementary techniques. So usually we use all our technique, and then we compare the results. In ramen, micro ramen are yes are important. Also, very very weak signal. It's not like in AI that usually you don't study too much, but here this is a strong, this is a medium, and this is very very weak. And we study also these. And so, just to have an idea, this is just part of the spectra of IR and microdrama spectra. It's a range. You can see different range, and so we compare really each band to the other, and then we do the segment as I've shown you just a few minutes before. Is easy? No. You have to be at the spectroscopy spectroscoper, so sorry, or Ramanist, and because in AI you can have some database or books. In micro Raman is quite difficult, yes, because the Raman instrumentation is very expensive, and so it's not all of the groups all around the world they have. So this is the main region. And so this is why you have to be a Ramanist. And then we have a study with the X-ray patterns. The X-ray patterns compared also with the C. So it's possible also to heat the samples and to do X-ray diffraction, do the heating. So we've done that from 35 degrees until heating, and also it is possible to do in this case with powder. We have not instrumentation to do with single X-ray diffraction. We do at room temperature, but I think it's enough. And also, we usually do also some micrographs, so the morphology of the crystals during this case, during the grinding. We have a study in this case, why don't if we grind the samples, because if you grind, yes, you have a PA, and then you grind, for example, with the other excipients. So you have to know what not only the shape, the morphology of your samples, but also what's going on, what's this transformation, what's going to transform it to one form to another. So at the beginning, it had this kind of shape that, and so also we measure the shape, and then we have studied the grinding sample, the grinded sample with the spectroscopic technique. Why spectroscopic technique? Because I was like, technique useful for identification, characterization of solid forms and of ligands and transformation of the samples. And so here in this case, we have a study when the sample was not grounded after 1 minute, after 5 minutes, after 10 minutes, and so on. And so, yes, it's possible to study the modification of the sample in during when it's grinded. Well, and then it is possible to study with DSC. So we have done also profile of all of these samples. And well, actually, a conclusion, what I want to see, I say that it is possible to study with Raman spectroscopy, but also with other different technique, and it's possible to study, and it's necessary to study not only with one, with IR, with Raman, and with X-ray diffraction, X-ray powder diffraction, and also with eating technique like DSC. I have not reported TGA, and all the technique useful for to study identify the solid forms, the solid form. Uh, Polymorphs or co-crystal or amorphous present, and you can, and then what I say usually at the end of my presentation, open also to collaborate with other. Well, actually, I work with a lot of group all around the world, in Europe, out of Europe, in India. I'm starting now in China, and so why? Because I believe in collaboration of the groups, and so I do my best in my work. And then this, for example, the other group, they send me the samples, or other groups do calculation, or we do calculation also. So we're starting to do calculation. And so, and these results are the results of quite a yes, of some years of our work, and they are not only our results, results also of collaboration of other different groups. And so I'm open also to your question if you have some. And so